So I'd like to kind of start off talking about automated data analysis, kind of where we're at with that. And the number one question I think that keeps people from running these algorithms, these black box algorithms that we don't know is how trustworthy is that data? And I'm sure that the answer is going to be depends somewhat. But maybe you guys could discuss. I think it's more trustworthy than most of our researchers think. But maybe you guys could talk a bit more about that. What you think?
I can start with that. Of course, the data you use for these automated analyses is as reliable as you make it reliable, of course, because the way we implement, for example, that kind of approach is in combination with a group of experts. For example, in a clinical application, it's easy to understand. You can join a group of experts in, for example, lymphomas, and they create those databases. So the reliability of the analysis based on those databases is only as good as the data you use to create them. Of course, if you do bad databases, the analysis will be bad, of course. So that's the point. And the other thing is it's not a black box because you can actually have access to those samples. So you can analyze those samples to understand what's inside. And it's not like for example, with a neural network that you don't know what's happening inside because it is what it is. So in that sense, and I'm not saying it's better, it's different because and that's a demand in a clinical application. You need to know, you need to be able to analyze what you are using to compare with. So that's the point.
Definitely good data in really makes a difference and to how it's going to work. Chris's?
Yeah.
You guys want to weigh in?
Yeah, I don't know. So you're right that it's all about good quality data. But I think the thing that people need to remember when they first approach it is it's not unsupervised. It's not standalone. There's a curation before, and there's some tweaking of parameters that may have to happen. And it's not just about throwing it into an algorithm. The data has to be well presented, high quality, and also you need to understand what some of the parameters are, which is where the documentation and attending seminars and talking to the creators really helps. Yeah. Talking to the bioinformaticians who really understand the maths behind it. As a biologist, I have a fundamental understanding of mathematics, but some of these algorithms are way beyond me. Yeah. And it's important that you liaise with the bioinformaticians properly.
Makes sense.
All right.
I agree with all those points.
Very good.
I really like starting with the idea that, as we say, garbage in, garbage out. Or I was working with some Spanish people and they said, entre basura, sale basura, right? It's cross languages, cross cultures. The importance of cleaning before you really start diving into trying to do automated analysis can't be understated. It's just so critical. So that was great to hear. I also like the principle of try to understand the components and why you're using them, and also just a basic way of understanding if they've worked well. There's pretty easy ways to understand if you get a total bad result out of t-SNE, for example. It might look like a ball. You have no heterogeneity that you're actually looking at. And that probably means, as was noted, that you have to tweak the settings a little bit, right? Understand some basic aspects, not necessarily of the math and how every algorithm works, but the general classes of algorithms, of which there's maybe like 6, and the general principles there. And then have just a basic understanding of, is this a sane result or not? And lean on us, lean on the people around you to help. I really like that. In terms of where the field is at, I think we have a lot of cool components that can be brought together to do a workflow that gets you to an automated result. Where I think the field is lacking in terms of what's out and available right now is dealing with really huge data sets. That requires a certain level of creativity. On OMIC, we're seeing people push into the billion event scale. in many places, right? With these large clinical data sets, for example. And I read a bioinformatics paper that comes out in 2020 and it says 800,000 events is an enormous data set. And it's just like, all right, there's clearly a disconnect here between what people are doing and what people are thinking in terms of who are developing these algorithms, right? So, my message to the field and where it's at is, certainly dealing with enormous data sets is almost going to be standard, really enormous data sets. And then another place that is sort of where the workflow ends, you may find interesting populations using things like clustering, dimension reduction, or other automated means. Labeling those populations is still a pretty human-centric step. At the end of the day, that's perhaps both from just a lack of available algorithms that can just canonically label populations, and we would just all agree on it. That's maybe because the algorithms don't exist, also because it's sort of a hard problem. I think there's some levels of disagreement and nuance in terms of how you should label a population. And so that is still sort of left to the human operator, and where I'd like to see some new innovations coming out and maybe some collective field action in terms of deciding how should we really label these populations. Let's just sort of decide on an official dictionary for it and have consensus on that. That'd be really cool. And then there can maybe be multiple algorithms that can fit you to that. So that's my snapshot on where the field's at.
Yeah, that was great input, guys. Sorry.
So to start to what Chris said, the metadata, the keywords is super important. So many people go on their flow cytometer and don't label their parameters. They sample 1, sample 2, sample 3. This is all completely useless. And you can't take your lab book and scan it and then apply it. You have to then do that by hand yourself. So when you acquire data, add every keyword, every parameter that you can think of, because it will save you a lot of effort down the line.
Yeah, great. So good data coming in is definitely a key point that everybody made in there. So kind of building off of that, you talked about kind of the future of automated analysis, where we need to go. Actually, it's a two-way street. Chris and Mario, you guys are obviously developing more tools that are better interface, things that non-biostatisticians can use. And Christopher, you're digging into R and writing your own algorithms that your standard flow or director or manager, we're not all doing that yet as much as we should. And so what do you guys think? Like, who needs, obviously it's going to be both sides need to be doing more. What can each side be doing more so that we can maybe reach some of these more nuanced R packages that aren't necessarily already rolled out into a nice, pretty software, and what needs to be done to kind of bring more data analysis to the common flow cytometrists?
I will support the vendors here, that they're generally doing quite a good job with OMIC and Cytobank and Venturi One, Flowdo, and FCS Express. They're all including a lot of these tools, and this is the best way to learn, because these are all kind of default implementations of these tools. And if you understand the output from these default implementations, then you can kind of progress onto to be able to alter them or use other implementations later on the line. There's no point putting into R if you don't understand the output. So using tools produced by these guys and by other vendors is a fantastic way to learn as well.
And I believe that it's important to, if I like to work in an environment, either using the Infinisite, OMIC, or whatever software, it's important to be able to bring those R algorithms inside my environment, so I can test them. And once I bring those results to my environment, the one I feel comfortable with, I can ask to the companies to implement a standard implementation of that kind of algorithm, and that's something important, because we need, from a company point of view, we may come up with new interesting tools, but they are only interesting if you can use them. And it's better to learn from you, implement a variation of that, learn with you, and then bring something more standard. So that kind of tools to have that connection to more open tools like R or MATLAB or something like that, it's very, very important.
Oh, great, that's good to hear. Chris, anything?
I sort of lost track of exactly what the thrust of the question was. At
Sorry,
first, I thought it was like, how could these tools be made better? But why don't we bring me back to tracking?
Yeah, so I guess the general idea of the question is, what can each side, the user of these algorithms and the creator, like the software engineers, you guys are doing a great job of bringing them to us in an accessible way. But does the user also have a responsibility in this as well? And what can be done better on both sides to bridge the gap?
Yeah, I guess I spoke to this briefly, but put in some cursory effort to understand what you're using before you maybe shove 100 million events into it or something like that, right? I'd say start small and grow from there. As your data set gets larger, as the complexity of your scientific questions get larger, trying to push these things through algorithms without having a firm understanding of them can be harder. So yeah, start small, grow from there. Reach out with targeted questions. I always like to see that. I don't want to hear about people struggling for a very long period of time when we could have, pointed them to a tutorial or answered some questions. And use clean data, spend some time on that, and publish your data sets. Be clear about exactly what the nature of the files are, the metadata that was brought up a second ago. That we always like to see that because we want to find good example data sets, et cetera, with clean metadata and a clear understanding of exactly how the files were cleaned or processed at all. And besides that, I feel like, I don't know, we owe more to the users, right? For package developers, I think vignettes and examples can always be better and things can always be simpler and more clear. So we're always working on that too.
There's a lot of undocumented features in a lot of packages that you said, right? But if you're new to it, if there's a website called Github that hosts lots of code, and if you just look for flow cytometry facilities or groups that heavily use flow cytometry, you can see their pipelines, their analysis workflows. That's a really good place to learn because this is how other people are analyzing their data, and you can see their code. And if you use FlowJo, when you use a lot of the R tools, you can click a button and it says output R, and it will actually give you the code FlowJo passed to R to produce the output. Because what FlowJo does for most of them is it just sends a command to R and receives the data. And it will actually tell you what the code is. And that's a really good way to learn as well.
Great. So really what I'm taking back from this is that us as users really need to dig in and learn what it is that we're using rather than running it and expecting output from it, which obviously makes a lot of sense. It's what we tell our users with our cytometers. So you bring up a good question about the publication of data and actually seeing what other groups are doing. Because we know there's a lot of variation and reproducibility of data, this could go a long way in standardizing the data that people get back out of it. Like, what should be published? Should the whole pipeline and the templates and right now raw data is kind of the standard, and then saying what you did, right? And then a lot of people will voluntarily put things up on GitHub, but I don't know that that's incredibly common that you see everybody's pipeline, the template of all of that. How much should as this becomes more the way we analyze huge data sets, how much should that be become more required by especially the larger journals or the specialized cytometry journals, right?
Well, this is predictable, but I would say it's absolutely of critical importance for even the simplest workflows. That's even if you take your basic cytometry workflow, which is probably compensating data and then you're scaling the data and then gating it and producing stats and visualizations and stuff. Those individual settings are so critical, and if you change them a bit, you can get wildly different results. And this has led to a lot of different problems with reproducibility in the field. At the very least, say what those critical settings are, down to if you're using by exponential scales, what is the width basis? What is the cofactor of your scales, et cetera? That information needs to be there. It's critical. So I'd say very necessary.
Mario, Chris, Christopher, you got any input into that? Like, what should we be requiring in journal submissions?
Yeah, so obviously the pipeline's useful. I mean, if you looked at any of mine, it's an absolute mess because your pipeline should be well commented as well. But I think the raw data is important because even in Flow repository, a lot of that data is curated. It's already been processed in some way, and actually having access to this raw data, especially with Spectral, because this has shown that there's some mixing can alter the data quite radically. And what people submit quite often is just the unmixed work. And we don't know what populations they chose. We don't know if there were cells or beads necessarily. I mean, it should be written in the manuscript. But having the actual raw data rather than some curated data would be the minimum, I would say.
Yeah, I totally agree. If you have very good data and you do an amazing analysis and then no one can take that raw data and obtain the same results, it's meaningless because you can say whatever you want. So it's important what you said about not messing with the raw data. Okay, you can do whatever you want to your data, but let me know what you've done to it and give me access to the raw data. Even if you have bad events that you take out, let me see them. Let me learn how to do that. How did you do that? So that's mandatory, of course.
Hopefully we see as we evolve in this, we see more of that stuff being published with our papers.
Some of it is learning. Actually, I find, in my education role, dealing with a lot of scientists, that a lot aren't even aware that cytometry is a sequence of steps in the first place. It's becoming more obvious as we're doing things more commonly, like cleaning and clustering and dimension reduction and stats, and the chain of things is getting longer. But to date, most people are used to just dragging their files into a software. The steps, those really critical, important steps, are happening in a certain order behind the scenes that you're not necessarily aware of the order that they're happening at all. And so it's sort of not the average person's fault. They're like, yeah, I just took the files and I gated them because that's what they think is happening, when in fact there's some critical steps that happen before that. So big learning lesson. There are these very important steps. All analysis is a sequence of steps. So yeah, I think once we all commit to that concept, like other fields do, then it should be naturally obvious what you'll see happening in the journals. And anytime someone does analysis, they'll be clear about those steps. So yeah, sorry to interrupt, but
no, that's great input. I really appreciate it. Let's take a question from the group. Cheyenne Yadav asked, they're working in flow cytometry. They want to learn a programming language. Which one should they pick?
Well, I guess I'll jump in. I answered this question. I say, unfortunately, the answer is R. Like, it's sort of obvious. It looks like Christopher agrees with me. R has become the de facto language for cytometry. That's not really related to the language itself. R is just a programming language. It's a tool, right? Like a hammer or a saw is a tool. But what people have done over the years is contribute, I don't know, maybe millions of hours, something incredible to the development of packages that go along with R. Then those packages are really what's doing all the amazing work, right? So you can do all sorts of different things in R just because that's where the packages are. And so that's why you should probably learn R and more importantly, how to use those packages. I agree with Christopher. There's some stuff in Python, but it's pretty incomplete. And MATLAB, there's some stuff in MATLAB as well. I don't like seeing that because of like the commercial limitations, that sort of thing. What you could focus on is just getting data out of FCS files. And that's pretty easy to do in multiple programming languages. And then you just have like a matrix of data, right? Just numbers. And then from there, it doesn't really matter what programming language you're using, but you do have to be aware of, you're probably not using a cytometry specific package anymore. Maybe you're using like scikit-learn or whatever, right? And so you could, for example, just parse data out of FCS files in Python and then pass that data around to the really amazing Python machine learning ecosystem. But just know that, they're not focusing on you as a cytometrist. However, when you're using R and you're using flow cytometry specific packages, they know you're a cytometrist a lot. And so you'll have documentation that's a little bit better for you, especially as you're learning. So that's my take on it.
I think Christopher and Mario, I see you made some
I totally
points
agree.
in the chat.
I totally agree. I don't know. I've done some things in Python, but if you are developing your own tools, you can use whatever language you are more comfortable with. But if you want to apply tools developed by the community, it's way easier to find good things in R right now than in any other package. So yeah, if you need to choose one, pick R for sure.
By the way, I want to clarify my previous point. The only reason I said it was unfortunate is because R is relatively challenging to learn as far as programming languages go. Yeah. It's incredible. It's one of the more impressive things maybe ever, the R programming language. But it's just, as a professional software engineer, I can say that still to this day, I'm confused and learning things about R, having spent, I can't even tell you how many hours working with that language. Whereas with some other language, I feel that I'm totally in control and mastered that language a long time ago with way less number of hours. So that's the only reason it's sort of a challenge.
I can vouch for R being tricky to learn as well. All right, so maybe I've got another question here. What is your opinion on how best to incorporate quality control metrics for automated data analysis? So what kind of QC files and QC data can we add in there to best incorporate?
Well, you would start with the normal ones. You could do your time versus fluorescence. You can do it automated in Flow Cut or Flow Clean or Flow QC in R, and you can do all these in FlowJo and FCX Express, I assume. You have to remove the extreme events. But apart from that, I don't know what other metrics we'd use, because I don't do any normalization. I will say that I've never done it. It's always been a bit of a black art to me, black side. I don't quite get it. But people who do it, because I don't do clinical data, so people who do it swear by it. And I think someone else should talk about normalization, but I believe it's very important.
Actually, that's what I wanted to mention is I've seen so many times people doing large studies and they'll get to a point, and let's just keep things simple. Let's not think about algorithms for a second or whatever. They'll get to a point and they'll say, is this change real? And we might say, I don't know, where are your controls? Like, can we look at the control? And they'll say, I don't have a control. Like, and then there's nothing you can do about that. So what I'd say is, especially anytime you have some sort of larger study, anytime you're collecting data in multiple collection events, maybe different sites, different time points, make sure you have a control that's just for batch control purposes. Ideally, it's some sort of tissue that's similar to the one that you're processing. Ideally, it's been processed in more or less the same way as your other samples that you're dealing with. If you want to throw in beads too, that's fine, whatever. Anything is better than nothing. But the question that you're going to have is, assuming that you're a B cell and you were processed on this day, approximately what should you look like, or any other cell type? And what we see often is that these things change just as a consequence of processing or reagents or instrument or whatever. And so if you lack the ability to understand that, you expect some cell type to be lower or higher expressing on a certain day, then you're just going to be wondering, is that real or a batch effect? So please, there's different products, I think, lyophilized cells, this sort of thing, but, experiment with it, practice with it. You want that control. You want to believe that it's really identical data and identical distributions between different collection events. I shouldn't say events, time points or anytime you're running the instrument and collecting data, right? So once you have that, you can choose to use it just to understand your data or to normalize your data, right? I agree normalization should be avoided if possible, but it is a solution that can be deployed with good effect. Actually, a question came up that I'll address now. How did I talked about normalization a second ago. That was an algorithm called the FDA norm. It falls into these one of two classes of normalization algorithms, one of which is just simply warping your data such that peaks align, and it doesn't use the concept of control, not like the one that I was just talking about. It removes batch effects, whether or not those batch effects are real, real biological differences or artificial differences because of how you collected the data, right? And that could be valid if you don't care about the relative level of expression of a marker, you just want them to align for phenotyping purposes. Okay, the other type of normalization, of course, is using actual batch controls that I just mentioned. Those create a model of how your batch controls change over different collection time points. And then it will use that model to change your data and fit it back to where it thinks it should be, right? Obviously, the topic is complex. If you're ever using normalization, make sure you understand why, as in you're looking at your data ahead of time, and then understand if it worked or not. Look at your data after, like down to the histogram peaks, and ask yourself, does this look like it worked? Because normalization can go wrong. It's not foolproof, just like none of these algorithms are. And you should supervise it in the sense of, you need to be watching what's happening with that algorithm.
I believe that we should start by trying to avoid the need for that kind of cleaning and normalization. And the first step is to standardize your sample processing, not leave the samples on the top of your table for a couple of weeks, and have your flow cytometer well calibrated and controlled over time. If you are sure that your samples are well processed and your flow cytometer is well calibrated, it's more likely that those differences you are seeing, or that population that shouldn't be there, it's a biological alteration and not an artifact. But I agree that even in those situations, there are variability, and you need to have access to those tools to clean the file, to normalize your data. And once you start analyzing the data, you need to be sure that it's a good set of data and not analyze bad information, of course.
Always good to reiterate, no bad data is
Yeah,
always
no
going to give you
bad
bad analysis.
Yeah.
And so we've got one more from Juana Gonzalez in the audience, is asking, what do you guys think of FlowSum as an analysis tool? I've used it. I thought it was really interesting. It's a good way to visualize some data. But it doesn't automatically analyze for you like everybody's saying. So what do you guys think?
As far as a clustering algorithm, it's probably the most commonly used of all time in terms of the number of people that have used it, in terms of its level of adoption within the community. That's probably because it has some really great documentation. It's an approachable and accessible package in R, right? Kind of talked about these points earlier. It also has really great performance properties. So it deals with larger data sets. This probably is more important from the perspective of if you're running on your laptop or something, it'll make sure that you can actually get your analysis done, as opposed to trying to analyze, like other clustering algorithms will top out quite quickly on just your average laptop, and your laptop might be running at 100% CPU for 20 hours or something like that. FlowSum doesn't do that, which is nice. So I'm going to look into a really cool project that I can't remember where it came out of, somewhere in Europe, is this method GigaSum. GigaSum is like a high performance computation version of FlowSum. So it's even, getting into these really cool high performance computing versions as well. Yeah, I'd say the mix of ease of accessibility and good documentation is good with FlowSum. It also has some good visualizations, as was said. I think they have some bolt-ons now that can do some cool, like differential statistics or comparative analysis. So that's cool. But usually what you'll have to do after you're running FlowSum is then decide what you do with those clusters in terms of qualifying them and visualizing any sort of differences between groups and batches and that sort of thing. Yeah, it's
Yep.
reliable.
Okay.
So I think
1
it's very
minute
good
left.
as well.
Sorry.
Yeah. I'll very quickly say then there's version 2 just came out very recently, and they've just released a Nature Protocols paper, which goes through step by step how to use it. And that goes to Chris's about the accessibility of it, which is why it's so popular. Some of the implementations and certain vendors' software is pretty poor because you can't move the nodes or gate them or anything like that. So you have to do it in R basically. But it's a great tool, and it produces biologically relevant information, which I think is why people like.
I can't thank you guys enough for those great talks and just a super informative discussion. I really appreciate it.